So I wanted to come and say a few words about EEGNet today. This is the data standardization with the EEGNet Open and Data and Analytics platform. EEGNet's a data and platform and analytics hub that we've launched since 2023, based at McGill University, but also part of the Global Brain Consortium and the Montreal Neurological Institute, and supported by the Ludmer Center, Brain Canada, Canary, as well as part of the Canadian Open Neuroscience Platform. So today I'll be talking about our data standardization project, the objectives and the achievements, a demonstration of the platform, and we certainly invite all of you to request an account if you have not already, and a bit of information on how to get started in terms of contributing data and tools, and how we can also support standardization efforts. So EEGNet is an open data platform and analytics hub. The goal of it is to provide open EEG BIDS analysis-ready data and to provide toolkits and online workflows to support advanced visualization with online head annotation and cross-validation of those hierarchical event descriptor tags. It also links to open source analytics so that researchers can run pipelines on the open data sets. And in total, with everything all together, at the end of the day, it provides an open platform for building those machine learning ready data sets, with the goal of making these available for translational applications in global health, truly worldwide. So to request an account, if you are not familiar with EEGNet already, please do go to eegnet.loris.ca, click request an account. You can also follow the link at the bottom to sign up for an account on CBrain, which is the high performance computing portal across the Compute Canada cluster. And that enables access to the suite of analytics tools. So in order to accomplish this, we combine the infrastructure of several different open source platforms built at the Montreal Neurological Institute. This includes the Data Hub, Loris, and an Analytics Hub, CBrain, which I've already mentioned again. And it combines these, in addition, with tools for preparing and standardizing data, set, EDF, and more modalities to come with a tool called EEG to BIDS, which I'll just be discussing a little bit more later. Altogether, this combines a data platform with those online workflows, supporting data sharing and linked with analytics to provide that support for gold standard annotated data that is ready for global applications. So since the project start in February 2021, we've rolled out the platform, the workflows, the data sharing, and the analytics, and we're currently in the phase of gathering more data sets and more tools and linking these across platforms. So this is in particular why I'll be inviting you at the end of this presentation to please get in touch about sharing your data sets. And as I'll also cover, we do support the preparation of data sets and standardization for, in EEG BIDS. So a word further about the infrastructure. Loris is the data hub, which supports the data storage and the workflows and annotation that it takes place on the EEGNet platform. CBrain is the sister platform for analytics, where the processing on standardized tools is launched. If you have any further questions, please don't hesitate. These platforms or these tools are deployed across many projects, including for the UK Biobank, including for Big Brain, including for many other projects you probably may have heard of already. So we were using a common infrastructure to deploy the EEGNet platform. This and technology was initially piloted for the MNI Open IEEG Atlas, which was spearheaded by Jean Gottman and Birgit Frauscher, starting in 2018, where we brought open intracranial signal together with visualization overlay on the anatomical aspect in an open online visualization engine based on Loris. And following that, in 2021, we built the IEEG Sleep Atlas for multimodal resting state and significantly longer recordings. In addition, this technology has been used for the Cuba Human Brain Mapping Project, which was the next chapter. in our EEG efforts at the Montreal Neurological Institute and the McGill Center for Integrative Neuroscience. This is a collaboration with Pedro Valdez Sosa to release 250 subject data sets on MRI and EEG BIDS standardized data with clinical and demographic data as well. And that is still an open project that's accessible to researchers as well. Following in that vein, this technology is also part of the Canadian Open Neuroscience Platform infrastructure, including the NeuroLibre publishing platform. To add it to that, EEGNet has also added data standardization toolkits, including mounted on CBrain, an open tool based on Brainstorm for automated EEG analysis. This enables open standardization of pre-processing pipelines with a templated boutique structure on the high-performance computing clusters. And we're also releasing notebooks that are going to support standardized study design analysis scripts. And this will be culminating in a public workshop in early 2025, enabling researchers to really take raw data to a process state that is ready for, that is also in BIDS format and ready for ingestion into EEGNet. So together with those tools, we also have a separate EEG to BIDS wizard, which is a standardization tool that works across any platform and is currently being released on EDF plus SET, coming soon this year. And the idea is that it makes the standardization process to the EEG BIDS format significantly easier, reduces the difficulty and the expertise required, particularly in terms of data structures or organizations. And it also compresses the data and makes it ready for import into any platform. So if you have any questions about how this tool interfaces, please don't hesitate to let us know. I'm also a member of the BIDS maintainers group, so we're keenly keeping updated with those standards. And as a result, EEGNet as a platform at a glance has released a number of open data sets and tools. Our users are continuing to grow, as are our papers and publications. And I thought I would end off on a little demonstration. So this is going to be a short video. And let me just see if I can get it to play.
To request access to EEGNet, click the link to request an account and register to receive an email with your login credentials. Remember to review and accept the terms of use before you log into EEG.
I'm sorry, I just want to check, is that audible to everyone?
Yes, it is good.
Okay, I'll continue playing, thanks.
EEGNet. The dashboard summarizes the data sharing and analytics resources and workflows available through the EEGNet platform hosted on Loris, an open source database built at the McGill Center for Integrative Neuroscience. Recent activity on EEGNet analytics tools mounted on the CBrain distributed processing portal are displayed in the top right corner. A visual breakdown of the datasets or projects gives different views of the composition and size of data available on the EEGNet platform. Click on the label of a dataset to learn more in the project information panel below, including publications, how to cite this dataset, and task descriptions. To view the EEG signal data, click through or use the menu bar to access the electrophysiology browser to view the session recordings from any project dataset, raw or derivative. All available recordings can be reviewed in the electrophysiology browser module. Search for the recordings of interest and select the dataset type in the links column, raw, derivative, or both. For a single session or visit, scroll down to view the signal plot and its interactive features, metadata, and a basic electrode map in 2D or 3D view. All downloadable files are available under the signal plot. When viewing one EEGNet recording at a time, You can use the interactive signal plot to review and edit recording events and tag specific events for all channels of a recording with specific head or score tags. Click Show Events panel to view all the events in the current viewing window. Press the I button to hide and show events. The Tag button shows any head tags associated with this particular event. Press the third button to edit the event. Hover over head tags to view additional information. You can add and remove head tags already used on this dataset and score artifact tags with this menu.
Thanks, everyone. I'll just mention in
To
addition,
request access to EEG,
I'll just mention that in addition, we've added channel selection, validation, and visualization of the channel selection since this video was made. So please do check it out for updates by going to eegnetlauras.ca and requesting an account. So to get involved, in addition to just accessing the platform, feel free to get in touch to prepare and share your data. As I mentioned, we have converter tools, uploader tools, templates for standardizing metadata, and containerizing tools on the high performance computing clusters. So the information for getting in touch with us is below. Particularly reach out if you'd like to spend time at our office hours where we can support data standardization efforts to EEG bids. So in conclusion, the EEGNet Open Data Platform and Analytics Hub houses, shares, prepares, and disseminates open EEG bids data sets that are analysis ready, provides the most advanced EEG visualization tool open, available, and out there. And it provides an online browser-based platform for head annotation and cross-validation for building these expert annotated data sets. It also provides open source analytics tools on high-performance computing clusters, in some to build machine learning ready annotated data sets for applications globally. I'd like to wrap up by thanking our supporters, our partners, and our global advisors, including our development team at the McGill Center for Integrative Neuroscience and the Ludmer Center. And with that, I think I will pass to our next speaker, Dora Hermes.
So I'll be talking a little bit more about the standards for electrophysiology annotations, and specifically hierarchical event descriptors, a specific library schema for EEG annotations called SCORE. And before I'm going to dig into the details of the methods, I would like to just describe a little bit about why I'm interested in this topic, because we're collecting large data set, and we generally only describe this in the method section with like one sentence, such as data were reviewed and segments with artifacts, or for example, we work with patients with epilepsy, so interictal activity were excluded from analyses. So that's all you see in the end publication about what's going on in the background. But what this doesn't capture is how does a lab annotate data across like years of data collection, or how does a collaborating site annotate data if you have data from multiple studies? And even like, are the published data actually annotated if you share data with a publication? And this becomes relevant because we're building a large data set for stimulation-driven connectivity, where we are using intracranial electrodes, and we're stimulating a site with a brief single pulse of 200 microseconds in duration. And then we assume that the signal spreads through white matter bundles, for example, to other brain regions, and then we measure the responses in other sites. And we are trying to do this across a large population of subjects from different ages. And we have a paper where we collected 74 subjects worth of data that were collected for more than 8 years. And they are all the way from 4 to 51 years old. And here you can see the examples of a brain rendering from a 4-year-old and a 38-year-old. And we looked at how, for example, you can start quantifying across these large groups of subjects For example, how age influences the speed of signal propagation. So all the data need to be carefully annotated, and any artifact needs to be removed or annotated or removed, because otherwise you're not going to have a large, consistent data set. So over more than 8 years, we had to systematically store these data systematically store the data, annotate the data, integrate the data. And then we can, for example, see that the speed and the latencies for younger people, for example, for superior longitudinal fasciculus, are longer than latencies for older people. And so how do we extend these data and enrich these data for reuse? And so we have worked with a center who collected this data for a long time at the UMC in Utrecht. For over 8 years, they collected these data and all sorted into bins. And now I'm at the Mayo Clinic now, so we're integrating these data across multiple centers as well, so that we can get different approaches. Surgeons often place electrodes in different ways across different centers. There's different approaches, so that enriches our knowledge of the human connectome by integrating these data across centers. And then we need to harmonize data collection as much as possible, but that's not always possible between centers, but BCI 2000 has sometimes helped us with that here. So for this type of harmonized data collection, electrophysiological data collection within and across centers, it becomes extremely crucial that we have standards to store the data and standards to annotate the data. And so that's what I would like to talk a little bit more about today. BIDs just briefly, just to recap, and then the standardized annotations using hierarchical event descriptors. With BIDs, if you have many different data types, such as, for example, the intracranial data, they're often combined with EEG data, MRI data, MEG data, and other data types. And even though BIDs was originally developed for fMRI, it's human and machine readable and should fit most use cases. And when I talked to Chris Gorolewski, he said it's actually a community effort, and there's people extending this to MEG, and you can extend it to EEG, and you can together with other people, and there's other people who are extending it to EEG as well. So it's a community effort. You can join this effort and work together to extend BIDs. And there's currently, for example, we extended to intracranial EEG. We collaborated closely with the EEG BIDs extension, so it's largely overlapping. There's current multi-electro array extensions. There's a stimulus extension. There's a PET has been accepted in BIDs now. And if there is some data that you have that you feel like are not able to store yet in BIDs or are not well validated, it is, again, a community effort. So please contribute, highlight your issues. And when we stored all these data in BIDs, we share them on OpenNeuro. But of course, they can be also stored on other types of platforms, and to share these data. So the data are shared in BIDs. They have hardware setup. They have the recording settings are all stored in BIDs. But what BIDs doesn't specify yet is how to share, for example, the artifacts, whether patients were asleep or awake, or whether there was epileptiform activity, whether subjects were moving or, for example, blinking their eyes, or whether that interrupts or whether the electrode popped or was bad. And so for those purposes, we're starting to use now and enrich these data sets further. We're using hierarchical event descriptors. And so we developed and library schemas are particularly useful because they allow you to develop specialized vocabularies with field-specific terms. For example, for EEG, sleep, artifact, and epilepsy annotations. And it follows the HED design principles, such that everything is machine readable, it's orthogonal, etc., and can be validated using the HED tools. And I'll also show how this is adopted and fully validated within BIDs. And so we worked on a collaborative effort in a hierarchical event with the HED-SCORE working group to develop a library for SCORE, which is standardized computer-based organized reporting for EEG. That was originally developed by Sandor Benickzy in 2017, and well described in their publications. We have a preprint archive on this topic. So in the original publication, they detailed many different types of features that you can annotate in data. For example, types of background activity, sleep and drowsiness, etc. And they're all really detailed described in these publications. And we had to make sure that those were fitting in the within the hierarchical event descriptors. And we just released SCORE version 2.0. And so they had to meet certain rules such as orthogonality, uniqueness, sparsity, and clarity. And this head schema is partnered with, this SCORE library is partnered with the head schema that, for example, has some standards for the artifacts incorporated that matched with SCORE, but also are potentially fit for other types of data, such as fMRI, etc., and are not just specific for EEG. If you're interested in looking at the terms that are used, you can surf to this website, and I made a brief video that shows you how to get there. So this is the head page, and I go through quick links to go to the head schema browser. And there you can see that we can see the head schema library, and I click on SCORE if I want to see the SCORE version 2.0. And there you can see that I have these different, you can see these different features from modulator, background activity, such as, for example, a mu rhythm. You can search also using search terms, such as if I want to, for example, see what types of interictal activity are annotated. I can click on interictal activity and see different types of epileptiform activity. And one of the things that I would like to highlight briefly in this within this video is that if I go back here, that you can see that all these terms, such as, for example, the epileptiform interictal activity, have a full description and also the sources. So it's fully visible where the original definition of these terms came from, so that if future updates are going to be available, for example, through the American League of Epilepsy or the International League for Epilepsies, and they're going to update their terms, we can see which terms are used here and which terms need to be updated, because these are large efforts from the International League Against Epilepsy to establish these terms and publish them. And now we can actually see where they came from, which years they were used, etc. And so, so the legacy is important here to see what the origin was of these different terms. And so after we have these, and now that we have these terms, we can start annotating different segments. So for example, here, there might be a clonic seizure. Before the seizure, there could be background activity. And after the seizures, there could be artifacts that can be annotated. For example, and in a bid structure, that looks like this. We can have an events file that has onsets and durations, which is required by bids, and then has these annotation types, for example, elec, LM, and eye movement, and channels. And the bids events file.json describes what type of artifact, what type of tags these are. And you can see here, for example, that the annotation type column is a head column. It has head tags, and IM is an eye movement artifact, elect is a non-biological artifact, a muscle artifact, etc., or a combination between these. And we can describe all these now. And that's not only that we can describe these, because what if we would make a typo somewhere here in the JSON file and we wouldn't know anymore what this would mean. But the BIDS validator actually validates this. So we're working on we have an example data set that will be in the BIDS examples. We're preparing to submit to the BIDS examples. And if you surf to the BIDS validator website, you can select a folder and, for example, see if your folder has any errors in it. And here in our example dataset file, you can see here that it's immediately tagged when there's a typo. So if you make a typo in some of these tags, it's detectable and it's machine readable, and it helps the annotator such that you're not accidentally tagging something that's wrong or that's not a specified term. And so the last point that I would like to make is that HED is already being used. So we just released version 2.0, and hopefully that'll soon be integrated in EEGNet. So I would like to thank Christine Rogers for the slide content. Here you can see that the head tags, for example, for artifacts are already being used. There's another preprint online about seizure score, where they also used head tags for annotating different data set, and we just released version 2.0. So
Thank
using
you.
BIDS,
Oh, sorry.
we can, so sorry, to summarize, using BIDS, we can use standardized structures and annotations for large data collection effort within and across sites. These can be used to share data, and with HED, we can add standard annotations, such as, for example, in the HED schema library for SCORE, as available in our preprint, and a schema is validated and openly available for science or for other use cases. And this is a use case for other schema libraries. So if you have another type of library that you would like to annotate, it can actually be imported in HED and fully supported and validated within BIDS as well. So thank you so much for my funding and all the collaborators and these community efforts involved in it. Thank you.
Thank you so much, Dora. I should have mentioned we have actually integrated the last release of HED into EEGNet since that came out earlier this summer. So
Oh, cool.
community
Exciting.
very much dovetailing. Absolutely. That's wonderful. So next we have a presentation from we have a video from Sandor, and Sam, you're going to play it, and you wanted me to check the audio and video. Is that correct?
Yeah, of course. Now let me just start.
Thank you so much for the kind invitation. It's a pleasure giving this talk about artificial intelligence for automated interpretation of routine EEG, the SCORE AI system. These are my disclosures. And this is the outline. I'm going to talk about development and validation of SCORE AI, and then I will talk about generalizability and external validation of SCORE AI, a project which we have just completed. Reading routine clinical EEG is much more than just spike detection. In skilled hands, EEG is an important diagnostic tool. Yet, it requires extensive training, not available everywhere. In resource-limited areas, there are no experts. And even in many areas in the United States, neurologists without fellowship training read EEGs. So no surprise, overreading EEG is the most common cause of misdiagnosing epilepsy. So there is a need for an automated interpretation of EEG, which provides clinically useful information at recording level. Now, what is clinically useful information at recording level? Well, first of all, you have to separate normal from abnormal recordings. And then you need to subclassify abnormal recordings into focal epileptiform, generalized epileptiform, focal slowing, and generalized slowing. This is much in line with the EEG classification proposed by Lüders, and this is also the primary input for the subsequent decisions on patient management. So, of course, you can describe the EEG at a higher granularity. But this, which we propose, these four classes, are sufficient to go on and decide on the next step concerning patient management, be the diagnosis or treatment. So we used the SCORE EEG system to train the AI model, and SCORE is the acronym for Standardized Computer-Based Organized Reporting of EEG. Let me show you how it works. So when you, the expert, spot an EEG abnormality, then you describe it using predefined items on the graphical user interface of SCORE. So here you can see this abnormality, and then you go to the SCORE graphical user interface, and then you click on the morphology, which is a sharp wave, and then this is in the right temporal area, and then time-related features. These are single discharges, 1 to 3 per minute. And then doing all these, you generate an automated report, which looks like this. But in the same time, you also feed these data into a database. And all these labels are linked to the specific points where they occur in the EEG. So this is an ideal basis for training an artificial intelligence model. So we did that, and we trained an AI model, which we named SCORE AI. Then we validated this on a representative data set of 100 EEGs. And the gold standard was the assessment of these EEGs by 11 experts who trained at different institutions. They were all board certified and they had experience with EEG reading. Now, here you can see the agreement among the experts concerning the EEGs. And you probably heard that before, that the agreement between experts when it comes to EEG reading is low, but that's not the whole truth. So those studies are usually based on assessment of 10, 15 seconds of EEG, but that's not the real world scenario. In the real world, you assess the whole EEG. And when we gave the whole EEG to the experts and asked them to classify these EEGs, then the inter-rater reliability, inter-rater agreement was pretty high. So concerning whether the EEG is normal or abnormal, then there was substantial inter-rater agreement. Also concerning the presence of focal epileptiform discharges. Concerning the generalized epileptiform discharges, there was an almost perfect agreement among the experts. Concerning the non-epileptiform diffuse abnormalities, the inter-rater agreement was again substantial, while the lowest inter-rater agreement was for the non-epileptiform focal, so for the focal slowing. This was high, moderate, getting close to a substantial, but not reaching the level of substantial agreement. And of course, when there were multiple abnormalities in the recording, then again, the inter-rater agreement was lower. Now, how does the consensus between the agreement between SCORE AI and the majority of the experts relate to the inter-expert agreement? Well, actually, SCORE AI came closer to the majority consensus agreement concerning two of these items. So for normal versus abnormal, the SCORE AI model came closer to the majority expert consensus than the average of the individual experts versus the majority. And the same was the case for the EEGs containing multiple types of abnormalities. And for the rest of the items, SCORE AI was as close to the majority consensus as the experts. Then we did a formal assessment of the diagnostic accuracy of SCORE AI, and we did not find any statistically significant difference between SCORE AI and the experts. Then we validated this on a single center data set from another Scandinavian hospital, but that hospital did not participate in training the model. So the EEGs from this hospital were not at all included into the training of the model. So we extrapolated the expected agreement between the model and the single experts from the large Norwegian center from the inter-rater agreement, which we calculated from the 11 experts. Now again, here you can see that for 4 items, SCORE AI came closer than what you would expect from the normal inter-rater agreement, and for 2 of the items, it was as close as expected from the normal human expert inter-rater agreement. But of course, the question is whether this is generalizable. Because when it comes to the application of artificial intelligence in medicine, there is a huge contrast between the large number of published papers and the few implementations in clinical practice. And this is largely due to the lack of generalizability and the over-optimistic results which are published. Why? Because many of these training samples are too small or not representative or from single centers. And then differences between various recording machines and patient populations are often ignored. And there is a lack of reliable external gold standard. And of course, there is a lack of well-conducted clinical validation studies. There is this culture as if the community was interested more in publishing papers than in real-world clinical implementations. So we conducted this external validation study in a geographically different patient population. This was from Montreal, Canada, from the MNI, from the data set of Birgit Frauscher and John Gottman. And we included an independent and balanced data set of consecutive patients. So based on the sensitivity and specificity which we measured in our previous studies, we expected that we should have at least 102 cases, but to make a balanced sub-classification between the 4 abnormality types, we had 52 abnormal EEGs, 13 in each category of abnormality, and then to balance this, we included 52 normal EEGs from different patients, of course. Well, importantly, this data set had different recording conditions with different equipment. In our previous study and in the development data set, we had Nicolet 1 EEGs. Montreal EEGs were recorded with Nihoe-Corten. They also had a different sampling frequency with 200 Hertz versus the 256, which we used in the development data set. And of course, different hardware filters. Very importantly, in this study, we used an external gold standard for all the categories. And these were derived from the electronic health records and the follow-up, which allowed us to benchmark, to compare the performance of SCORE AI with the performance of the experts. In the previous study, the experts were the gold standard, but here the experts served as benchmarking, as a comparison for the performance of SCORE AI, because the gold standard was external. Of course, all analysis was done blinded. And very importantly, we used a fixed and frozen AI. So first, we assessed whether the sampling frequency would affect the AI model or not. How we did it? Well, we started by using the dataset from our previous study. And first, we downsampled them to 200 Hertz, then we upsampled them, sampled them back to 250, and we checked whether this affects the performance. And it did not. So here you can see that the performance is unchanged. This is the dataset of 100 EEGs, which was scored by the 11 experts. And then this is the large single center dataset from Oslo, from Norway. And here again, the downsampling and upsampling did not affect the performance. So we were confident that 200 Hertz would also work for the Montreal data. And then the next issue we had to solve was how to bridge the difference between an EEG result and the diagnosis. So the gold standard is a diagnosis that comes from the medical charts, but the EEG is a class. So how do we bridge these two? Well, we used pre-established rules for both the experts, the 3 experts who served as a benchmarking, and the AI model. And these are based on what we do in the clinical practice. So for generalized epilepsies, we said that generalized epileptiform EEG abnormality must be identified in the recording. However, we know that many patients with generalized epilepsy can also have focal abnormalities. So we allowed also for focal epileptiform and non-epileptiform abnormalities too. But generalized were mandatory. And for focal epilepsy diagnosis, of course, presence of focal epileptiform EEG abnormality was mandatory, but we also allowed for focal non-epileptiform abnormalities. And in the same way for a diagnosis of focal non-epileptiform, then focal non-epileptiform EEG abnormality was mandatory, but we also allowed for diffuse non-epileptiform abnormalities. And for the diagnosis of diffuse non-epileptiform, then we said it's mandatory to have a diffuse non-epileptiform EEG abnormality, but we also allowed for focal non-epileptiform EEG abnormalities as well. So here you can see an example of a focal epileptiform abnormality. Here you can see an example of a generalized epileptiform abnormality. This is an example of a focal slowing, and then this is an example of a diffuse slowing. Now, concerning the inter-rater agreement, the inter-rater reliability, we reproduced pretty much the same what we saw in our previous study. So again, if you give the experts the whole EEG, then they agree about the main classification of that EEG. So here you can see that there was substantial agreement for normal versus abnormal among experts, also for the focal epileptiform abnormality, for the diffuse non-epileptiform abnormality. And then it was almost perfect for the generalized epileptiform abnormality, but lower in the high moderate level, approaching substantial, but not reaching that. So high moderate for the focal non-epileptiform abnormalities. And then the agreement between SCORE AI and the experts was again in the same range. There was no significant difference. Now, let's compare the experts with the external gold standard, and let's compare the SCORE AI model with the external gold standard, and then let's do a benchmarking between them. And this is a busy slide with many tables, but here let me guide you through this. So on the top here you can see plotted the sensitivity, here the specificity, and here the accuracy. And then in red, you can see the performance of the SCORE AI model, and blue, the experts. And you can see that for all these diagnostic accuracy categories, they perform in the same way. There was no statistically significant difference between the performance of the experts and the performance of the SCORE AI model, neither for sensitivity, for specificity, or for accuracy. How about sleep and normal variance? Well, 11 of the 52 normal EEGs had non-REM sleep, and the fast classification during awake and the sleep was at the same level. And then we had 14 of the 52 normal EEGs containing normal variance, 6 sharp transients, wicked spikes, 3 small sharp spikes, 3 EEGNet rhythm, 1 rhythmic mid-temporal theta of drowsiness. Well, unexpectedly, because this is rare, but we also had 1 SREDA, subclinical rhythmic electric discharge of the adult, and there was 1 EEG with a temporal slowing of the elderly. But false classification by the AI and the experts was again at the same level when it came to the normal variance as well. Now, SCORE AI has been integrated into the NATUS NeuroWorks system, and it's called AutoScore. How it works? Well, it scans the recording and then it highlights where you can see the screenshots, the 16 seconds epochs, where you can see, for example, an epileptiform focal abnormality. And then if you click on one of these examples, the EEG jumps there and presents you so that you can either confirm or delete these markings. Now, how do you roll this out to the resource-limited areas where there are no EEG machines, no EEG equipment available? Well, we tried to address that problem as well. together with the Technical University of Denmark, we developed and validated a very inexpensive EEG system, which uses a smartphone or a tablet for processing the data, and then it uploads them to a secure cloud storage. Very importantly, because in the resource-limited areas, there are no technicians who could do a reasonable quality EEG. We also developed a simple app which helps the inexperienced users, the inexperienced nurses or social workers to record a good quality EEG. So here you can see the graphical user interface output from the smartphone, which guides the inexperienced user through the steps of EEG recording and helps the inexperienced user to make an EEG of a reasonably good quality. And here you can see that we continuously measure the impedance, the signal quality, which was really good. So we hope that combining these two things, this inexpensive EEG and the automated EEG interpretation based on artificial intelligence, we have bridged the diagnostic and therapeutic gap also in the resource-limited settings. In the end, I would like to thank my colleagues and friends who contributed to these studies. Thank you for your attention.
Thanks, everyone. Especially thanks to Sandor for that contribution. It's great to see work building on data that we've shared, open, that the standards have been defined through efforts of Dora and her colleagues, and of course Sandor, and that Sandor's group has of course been bootstrapping for more applications and integrations and more platforms. So I think it's a very interesting time to see all these efforts coming together for standardization and for the really rapid application of automation for standardization. As Sandor mentioned, there's a lot of ways to watch this carefully and things to look for, but it's important so that it builds on the community efforts for standards such as HED and the new release of SCORE. And I think we're going to see some more rapid development from here. Did we have any questions from the audience or anything anyone would like to come back to from the slides? Dora, we can absolutely continue talking about the fantastic news of the new release of SCORE. So that's SCORE 2.0. In addition, HED came out with version, I'm going to say 2.8. Did I get the numbers right there? Earlier this summer, which EEGNet's always already integrated. And so I guess that one of the questions could be what's next. We go and we kind of apply this on the platforms and we keep going from there, get community feedback, and what else? Papers, I'm sure.
Yeah, I think one of the things too, you're asking about like what's next. I think there's like many things that you know, in terms of the library schemas, there's many different options. People testing things out, battle testing things out on data sets will actually help a lot. There's a lot of like when you start to put your example data sets together, there's a lot of issues you start running into, or like small things, no major things, but like some little things that you think like, oh, I don't know how this works. Clarification, the same with bids, like how do I actually use this in bids is, you know, are things that, you know, people are, you know, it would be really exciting to see how people are going to start testing this and then combining it with AI, I think, to see if we can automatically attach annotations. Will take off as well. One of the things that is important, for example, is that there's in bids, there's more and more standards for how to add annotations. Within the stimulus BEP, you'll see there's a new BEP 44 for stimulus configurations, where people can more explicitly describe what type of stimuli were shown or what happened, for example, during a movie or in, and so I think there's a lot of efforts that are open for contributions that are going on. I think, yeah.
Yeah, absolutely. I think the descriptive ability of bids is the enrichment there is getting much, much stronger, not just in the stimulus BEP, but also in the provenance in development. I think many people in the community are aware that there was sort of a shift in 2023 between bids, raw and derivative conceptualizations, such that really the richer provenance description is kind of more fundamental to where the direction that we're going with considering something a derivative. But I think there's plenty of room for discussion, and I think that enrichment certainly supports many things going forwards in terms of supporting quality analyses and things people do with the data down the line. And I see Robert here. I'm going to just invite you on stage, Robert. Please take it away. We'd love to hear your thoughts and questions and comments.
Thanks. Thanks for the very nice presentations. I was so that's, it's really nice to see that this is already being incorporated in a commercial product, making it like much more widely available. But I'm also wondering, like, which stakeholders would you like to have on board, and do you not have on board yet? So where are the areas that we as a community should be working towards to try to get like more people exploiting this, contributing to this, disseminating this, etc.
Great question.
And I think it's mainly a question for Dora, because I'm mainly thinking like how do we make clinical and societal impact with this, which is of course through the like, for example, through the implementation of SCORE.
Ooh, it depends on the so I work with a lot of clinicians. I'm not a clinician myself. So some of these packages can be expensive, and not all hospitals have the funding to, for example, incorporate some of these automated tools. And that's one of the questions I had for Sandor, who's not here, is like, can to what extent is this possible? Like, what's the, for example, for the EEG, the EEG low cost version, how much, is that feasible for people? So I think there's two types of things that come together in this field. One is from the researchers giving like specific new tools to be able to use. Making those openly available will be extremely essential for future impact. But most hospitals have clinical systems. Those software systems have to be entirely validated for clinical use. So before they can use it, it needs to be in a company, in a certain system, before the clinicians feel comfortable using it. Before that, it's like this currently it's the interaction between the two that really drives the field forward. And some of the new sites that are testing to what extent the clinical annotation works well with the if there's a new software system, does it work? To what extent do they feel comfortable using it? Is it maybe giving a first look at things? But we need things pretty much available, and it's difficult for companies to adopt, to do these adoptions, or there's a switch to that, with people being more comfortable to use an AI, for example, as a preliminary tool or as a first look. But there are still people reading the EEGs, so it's a combination between the two things. But yeah, things are coming together, I think, in that field.
I'll also add that in terms of other things being available and engaging more stakeholders, we are always looking to drive more data sets into sort of the shared ecosystem. So standardized and scalable effectively with large ends and with large amounts of coordination. So I think that will also be really interesting for input to AIs and also for testing outputs of AIs. And that's particularly why that outreach is really important, I think, right now. Yeah.
Okay, thanks.
Yeah, in terms of bids and standardization, so so we have this NEMAR database that's a collaboration with Open Neuro, and there is a 365 bits dataset, which means experiment stored. And we've learned a lot over the past couple of years, and we'd argue for stronger standardization rather than weaker one, because in our experience, about 70% of these datasets are very difficult to use, and probably at least 30% completely unusable. So that's food for thought. And so I think stronger standardization in the future in terms of what's in the data. You can just dump data, but most of the time it's not super useful.
So more specific definitions and
It's more enforcement. Lots of the data that's submitted is just corrupted. In the sense people forgot to put events, or they forgot to put the channel locations. So, you know, if you have 64 channels and your channels are 1, 2, 3, 4, 5, 6, there's nothing anybody can do with the data. And you can always contact the offers. In our experience, we contacted the offer every time there was a problem, but they very rarely respond because, yeah, people are busy. Yeah, so a lot of things can be done. Or if you have events 1, 2, 3, 4, and you don't define what are these events, and very often people, you know, even if they put the paper, how are you going to do with the paper and these events 1, 2, 3, 4 to reconcile the fact, okay, this is the oddball, this is the standard or something. And, you know, very often it's much more complicated than that. So I think, yeah, it's not like there's a magic bullet, oh, we do this and it's all fixed. It's more food for thoughts. And we're actually preparing a paper with detailing the content of NEMAR, so the 365 datasets, how many datasets of this type, how many datasets of this type, how many can be processed, how many are data epochs, how many are events, are blocks, block design, you know, eyes open, eyes closed. So that's hopefully going to be helpful to move the field forward in terms of stronger standardization. And of course, you don't want to be too strong because otherwise people won't even submit their data. But there's still things you can do so the data is reusable. I think reusability is key.